Let's go for the first part of session 4. Okay, I really enjoy doing different sort of water activities, including paddle boarding and using paddle boards on the lakes outside the town. We have been doing the paddle boarding with a group of my friends outside, and we have been enjoying the time there. And when we have been doing the paddle boarding, somebody just came and with the boat, and he was just basically passing the process, or sorry, she was passing us. And she told us, okay, gentlemen, you are enjoying your time. Just be careful about cotton mouse. And we have been, what is cotton mouse? And then we came out from the water and checked the cotton mouse. And cotton mouse is an interesting snake that is swimming across the lakes here in the state of Oklahoma. When he opens his mouth or her mouth, you can see there is something like a white spot in the mouth. So he's famous for being under the name of cotton spot or cotton mouse snake. And the snake is poisonous. And after realizing that, the entire experience of paddle boarding over the lake changed, because we have been, especially for the first few times that we have been doing that after being exposed to that information, we have been checking, is there any snake over the surface of the lake? So you can see the visual experiences of trying to find a snake over the seas of a lake is an interesting cognitive experience. How you are careful right now in terms of, is there any movement that is related to a snake over the lake? And the visual processing of finding a snake over the lake is going to be something interesting. So it's something like a personal experience. I'm not sure, have you ever experienced this sort of information that changed the way that you see the environment around you? Okay, and we are going to talk about those type of cognitive processes that are going to change how we see the information that is coming from the environment and the periphery, and how it's going to being processed inside the brain. And it's quite interesting that the snakes have had a significant effect in the vision system of human beings and primates. And there are discussions. There's a specific book that might be interesting for you if you like to read the book. And the book by Lynn Isbell is talking about how primates and human vision system is being developed mainly because of having snakes in the environment. Because of the really important role of snakes in the way that we could survive, or they could basically poison us and then make lots of health consequences for human beings. So we over the time, it seems that human being or primates basically try to evolve their vision system to be able to detect the snakes inside the forest or inside the different fields. And it seems that we have a sort of co-evolution with the snakes. And the snakes try to be better, and then the primates try to have a better vision system. And even there is a discussion right now that the quality of visual system that we already have as human beings is far beyond what we usually need in the daily life that we have. And the reason that we have developed such a really sharp, high quality vision system is able to detect really small movements in the environment is that we have been in a co-evolution with the snakes to be able to develop these really complex visual system. So that is quite interesting, that how our vision system is being affected by environment and how we have such a complex vision system. And we are going to discuss about those characteristics in the visual system in human brain. Let's start with a simple question in terms of, okay, we have a visual scene. And when we see a specific visual scene, how can we start to process that? What are the items that should be processed for us to understand what is happening in the environment around us. Okay? So what are the visual elements that should be processed when we are seeing a scene like this? I want you to kind of open up your chat space and just write down the elements in this picture or photo that is going to be processed, that should be processed to give you a complete sense about what is happening in this scene. What are the things that should be processed here? Just kind of use your chat space and write down the processes that you think that are important for processing the scene that you already have in front of you. Okay, you can see that we have lines. So we basically have lots of lines. Okay, so we need to be able to perceive these lines in the environment to be able to see what is happening. So we have lots of lines. Okay, we have different sorts of colors, different shades of colors. Okay. Then we have different objects. We have different sorts of objects in the environment. We need to process those objects. And then we need to also see how they are related to each other. So one object to the other object, where these objects are located in the environment. And probably after processing all those things, we will start to kind of relate that to our memory or other things to be able to have an understanding what is happening here. So there are different levels of information that should be processed when we see a scene to be able to have an understanding about what is happening inside the scene. If I bring up another picture which has another scene, still with hands and instruments and an action that is happening here. So we are talking about an action. And even like the previous kind of picture, you might have even a sense of movement. You might feel that, okay, this gentleman is kind of cutting out something here with a specific direction in the scene. And you have the same sort of feeling of an action that is happening and even the emotion that is going on right now, and the fire that you have under the spoon. Those who are familiar with these kind of scenes, they know that is a person who is cooking heroin or preparing heroin for injection. So that is the way that usually people prepare heroin for injection. And when you know that information, then you would start to have another level of information in this scene. So beyond the basic visual processing that we have in this scene, there is a specific meaning to what we see here. And that meaning is important. And the meaning is also to define how we are processing different pieces of this information. So we need to start to process all these minor pieces of this information here in the scene. Meanwhile, we need to also, the meaning would change how we come back again and pay attention to specific pieces that meaning is going to kind of attract attention to those pieces. So it's something like a feed forward and feedback processes going on together. So I will discuss about those minor details, okay? And when I want to compare these two scenes from each other, a scene that is related to kind of a normal, routine, healthy behaviors, and a scene that is related to drug use, okay? Technically, what we have in those scenes, if I just make something like a conjunction between these two kind of scenes, what are the factors that are shared in terms of the processing? In both sides, we have processing of line, we have processing of color. All these things are the same in both shapes. But then we talk about what are the things that we have here and we do not have here, what are the processes that we have that are involved when we see these pictures. And especially when we talk about specific groups, like those who are already using heroin. When a person who is already using heroin, if we just compare them in terms of the processes that are happening, what are the brain areas that are involved in these processes? So I'm going to give you some kind of more information about how these processes are happening inside the brain. To be able to do that, as we have discussed before, last session, we discussed about fMRI, and we discussed about how we can have better signal in fMRI. And we discussed if we want to have better signal, we need to have multiple stimuli. We need to have multiple stimuli to be able to have enough, let's say, bold response. If we want to have a really good bold response, we need to have multiple. It is not going to happen with just one stimuli. So we need to have multiple stimuli in blocks. And we need to repeat that again and again and again to get sure that it is not just noise. We need to get sure it is a real signal that is happening. So we need to repeat this several times. So if you want to do that, we need to have a large database of pictures to be able to show these pictures inside the scan to people, and then let them kind of process that and then do the fMRI to see what is happening inside their brain. So we recently developed a large database of pictures to be able to show these pictures to people and then record what is happening inside the brain. But the challenge is, okay, what are the factors in these images, in these photos, that should be considered when we select these photos and put them inside a database? What are the factors that are going to be important for us? Because we technically like to get sure that the basic information processes are going to be the same for both drug-related and neutral pictures. So we need to control them. For example, here, you can see two sets of pictures. One set, the left one, as you can see here, let me just bring in, okay. As you can see here in the left side, these are the opioid-related pictures, okay. This is picture 1, 2, 3, 4, 5, 6. So those 6 pictures that we are showing people, okay. And we repeat that 4 times. And then we have neutral pictures with the same idea, okay? What we need to have, we need to get sure that the basic visual processing that are happening for drug-related pictures are going to be similar with the neutral pictures. If we have hands here, we need to have hands on the other side. If we have tools here, we need to have tools on the other side. If we have hands and tools, we need to have hands and tools on the other side. And if we have faces, we need to have faces. Otherwise, if we just compare the photos that already have faces to photos that do not have faces, you will have activations in the areas that are not interesting for you. Because here, we are interested to the activations related to, specifically to what we call drug or drug craving, compared to just basic visual processing that are happening here. So what are the factors that are going to be important? First of all, as you can imagine here, it's important that overall we need to have the same level of brightness. We need to have the same level of contrast. We need to have the same level of objects that we have inside this environment. Otherwise, if we do not control for these factors, then we will have activations in the areas that are not as important as what we have been planning for before, okay? And we need to do that for other drugs to be able to control them. For example, this one is for methamphetamine. So we have different sets of images. But the idea is the same. So we would really like to have the same level of contrast, hue, the color, everything that is going to be almost the same or controlled for these factors. Otherwise, there are going to be problematic. And then, as you can see here, we have control for even between meth and heroin or opioid. And then we developed 3 sets. Why? Because we have been interested to show these pictures to the people before an intervention, after an intervention, and then in follow-up. Okay? If we show them the same pictures that we have been showing them in the before and after, then we'll have a kind of strong memory effect. That would make things a little bit complex. But we have been interested to show distinct sets of cues in different time points. So we developed a larger database of these visual cues. What are the areas that we are interested to see inside the brain? Areas related to executive control. And these are the functions that we'll discuss about them in the next session. So we'll discuss about all these items one by one in the next sessions. We are interested to know about executive circuits, salient circuits, self-directed circuits, memory circuits, habit circuits, gain and loss and punishment circuits. Okay, these are the circuits that we are interested to. But technically, we are not very much interested. This is the occipital cortex. So as you can see, here is the occipital cortex. Okay, so this is the medial view, this is the lateral view, this is the occipital cortex. As you can see here, none of these networks that we are interested to is located in the occipital cortex. Why? Because we try to control for those kind of vision processing related information between these two sets. And what we do, we subtract. We basically measure what is happening in drug related picture minus what is happening in the neutral related pictures. As we are expecting to have the same visual processing for drug and neutral pictures, what we have, this is the final result of that kind of contrast when we make a kind of minus between drug and neutral, okay? And as we are expecting that the level of visual processing that are happening in the occipital cortex in both sides are going to be equal, we think that that is going to be canceled out, okay? So we are not expecting to have any activation in the visual cortex. True? Okay. This is the activation map that we have in an actual group of subjects, 65 MET users that we showed those pictures, and this is the activation map. What is the section that we have here? The section is, and you can write it in the chat space. What is the section that we are seeing right now? It is an axial or transverse section, okay? So here you can see that in the occipital cortex, this is the front, this is front, this is back, okay? These are eyes, I mean eyes are here. So these are eyes. Front and then back. This is the occipital cortex. So good. We can see that there is not very much activation in the occipital cortex. Even there are some what we call deactivations. It seems that the activation is even lower in here is drug more than neutral. Drug versus neutral contrast, okay? But we have lots of activations in areas a little bit ahead in the visual cortex. So it's interesting that we have some deactivations in just the cone of the occipital cortex, but we have significant level of activation in the anterior parts. Why? This is what we have not been expecting before. Why we have activations here? So we developed a large database. We have done a really good job in controlling for those factors. Even for those photos that I showed you before, those photos we have published a database. You can find a database. In the database, we have provided all the psychophysics features of these photos one by one. The level of craving, the level of processes, the familiarity of these items, and we control for all those factors, okay? Even after controlling for all those factors, what we can see here is a little bit upsetting. Why we have activation in the visual cortex? Why do we have activations in visual cortex? And then, if I want to just summarize the results that we have, if I put them in the brainotome ROIs, if you remember brainotome atlas, we discussed about brainotome atlas in the last sessions in details. If I make an average of these activations in the brainotome ROIs, this is what I'm going to see. Here is what you can see here in the brainotome atlas. So there are activations and deactivations in, this is the frontal area. If you remember the abbreviation, superior frontal gyrus, middle frontal gyrus, inferior frontal. So this is basically the frontal part. And these bars are showing the average signal that we have in these ROIs. So we basically measure the level of signal that we have in these ROIs and demonstrate or presented the results here, as you can see here, okay? So we have specific activations here, but here what we can see, this is the lateral occipital cortex. There are areas in the lateral occipital cortex that you have a really high level of activations here. Why? Why do we have these activations here. Okay? So that's a really interesting and important question. Why do we have activations here? So I knew that we will have activation in the ventromedial prefrontal cortex. So these are the ventromedial, basically these are the VMPFC areas, okay? We have activations in inferior frontal gyrus here. Here are the areas that you can see. Inferior frontal, some activation in the inferior frontal gyrus. Even some deactivations in middle frontal gyrus. This is the DLPFC, DLPFC. So there are some inactivations in DLPFC here, okay? As you can see here, there are some deactivations here. And it's interesting that there are some activations on the, as you can see here, on the left. This is the left frontopolar area. This is the frontopolar area, the left one. And these left frontopolar activations are connected to the VMPFC activations. So those who are interested to the brain stimulation, they can stimulate left frontopolar area with the hope to be able to modify the activations in the ventromedial prefrontal cortex, and that is going to work. So we have all these activations, and these are kind of reasonable. So we even have activation in the kind of striatum, the things that are interesting to be seen. But why do we have activations here? So that is what we are interested to explore and think about together in the next steps, okay? So that is one question. So I have started with two questions. One about the snakes, that we have started to see why we are kind of seeing snakes in a really high quality, even a small movement in the kind of grass, when you are ready to see a snake, could be something alarming. So you can pay attention and see that in your peripheral individual processing. So how the visual system is working on that way. Or in the fMRI kind of experiment that I discussed with you, how we have activations in visual cortex in a way that we have not been expecting about before. And there are some other kind of questions regarding the visual processing. For example, we have, I'm not sure how many of these visual illusions have you seen, but there are lots of visual illusions. If you see a scene like this is a static image, so it is not moving at all. It's just a kind of 2D static image. Okay? If you start to see it from the center, just see it for 30 seconds and tell me what you are seeing here. Do you feel that the grooves and humps are fading to each other? So you have a feeling of motion and also changing between groove and hump, and just they change to each other. So you see how these things are interacting. This visual hallucination is being developed by vision scientists in University of Surrey. As you can see, Dr. Piana. And so he basically developed this visual hallucination, and there are more details about that. I'm not going to go to the details, but it's quite interesting to know where he is located in this part of Italy. And then we have another visual hallucination, illusion. What do you see here? How many people here are seeing these sort of snake-like kind of spirals are moving? Do you see the movements? But technically it is a static image, so it's not like an animation, just a static image. How this is happening, how these movements are happening in the periphery without having a real movement, why these things are working like this. So it is another kind of international scientist from Japan, from Osaka, Akiyoshi Kitaoka, from universities in Osaka. And you can find his web page. He has lots of really interesting kind of visual hallucinations in his web page, and he's discussing about why these things are happening inside the brain. And he has a really active kind of Twitter channel as well, so you might like to follow him in Twitter if you are interested to the visual illusions and how these visual illusions are happening. So these are the questions: why we have visual illusions, why these visual illusions happen, and how the visual system could be tricked by these simple kind of visual stimuli, and why they are behaving like this. Okay, just see this picture for a small kind of time. What was the difference between the right and left one? Let me just bring them back to the right position. Can you see the difference better right now? Why? And this is the same photo. Now you have a kind of another level of perception from what is happening in the photo. Why we are not really well in detecting such a kind of significant change in the face of this really nice gentleman, and we cannot see that when the picture is top down or basically bottom up. To just change the flipped in the photo. So why we have this significant difference when we see the picture from the other side? Why the visual system is working like this? So these are some of the major questions that I'm trying to address to some extent, and just keep the floor open for those who are interested to know more about the visual processing and how the vision processing, the visual processing are happening inside the brain. So we'll discuss about some of these technical details in the next steps.